might have heard about the importance of proteinaceous diet. Have you ever thought about the chemical structure of proteins? What are they made of? In this class, we will learn about proteins and their building blocks. The word protein comes from Greek word proteos, which means of prime importance. Proteins are polymers of amino acids. Amino acids are molecules which contain amino and carboxyl functional groups. Amino acids can be classified as alpha, beta, gamma and delta, etc. depending upon the relative position of amino group with respect to carboxyl group. As displayed here, alpha amino acids contain an amino group attached to alpha carbon atom. Here, R represents side chain. All proteins are polymers of alpha amino acids. There are 20 alpha amino acids present in nature. I have listed a few here. They combine in different ways to form different proteins. Amino acids are generally represented by a 3 or 1 letter symbol. All of them also have trivial names derived from their property or even source. For instance, tyrosine is so named because it was first obtained from cheese. This name is derived from the Greek word tyros, which means cheese. Amino acids are classified as essential and non-essential. Those amino acids which can be synthesized in the body are known as non-essential amino acids, while those which cannot be synthesized in the body and must be obtained through diet are essential amino acids. Here, tyrosine is a non-essential and methionine is an essential amino acid. Amino acids can be acidic, basic or neutral. Neutral amino acids contain an equal number of amino and carboxyl groups. If the number of amino groups are more than carboxyl groups, the amino acid would be basic, and if there are more carboxyl groups as compared to amino groups, this makes amino acid acidic. Now, we will study the physical properties of amino acids. Amino acids are usually colorless, crystalline, water-soluble solids with high melting point. They behave like salts rather than simple amines or carboxylic acids. In aqueous solution, the carboxyl group of amino acid can lose a proton and amino group can accept a proton. This gives a dipolar structure to amino acid known as Zwitter-ion. These Zwitter-ions are electrically neutral and exhibit amphoteric behavior. Looking into the optical activity of amino acids, you can see all naturally occurring alpha amino acids except glycine are optically active. The naturally occurring alpha amino acids exist in D and L forms. Most of them have L configuration represented by writing the NH2 group on left hand side. As mentioned earlier, proteins are polymers of different amino acids. Here you can see the condensation polymerization of glycine and alanine to form glycylalanine. A peptide bond is formed when carboxyl group interacts with an amino group. The product of this reaction is called a dipeptide because it is made up of 2 amino acids. If 3 amino acids combine, then the product is called tripeptide. Thus, depending upon the number of amino acids linked, it is called tetrapeptide, pentapeptide, etc. When the number of such amino acids is more than 10, then the products are called polypeptides. A polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 unit is called a protein. Next, we will look at the classification of proteins. On the basis of their molecular shape, proteins are broadly classified into fibrous proteins and globular proteins. Fibrous proteins are long thread-like proteins which run parallel. They are held together by hydrogen bonds and disulfide linkages. Due to the presence of such strong interactions, these are generally insoluble in water. Common examples are keratin found in hair, nails, fibroin in silk, myosin present in muscles, etc. Globular proteins result when chains of polypeptides coil around to give a spherical shape, and these are water soluble. Globular proteins maintain and regulate life processes. For instance, insulin regulates blood sugar level, right? Other examples are albumin, hemoglobin, etc. Now let us discuss the three-dimensional structure of proteins. In the descending order of their complexity, proteins can have primary, secondary, tertiary and quaternary structure. Here you can see the amino acid sequence starting at N-terminus from methionine and ending with leucine at C-terminus. Such specific sequence of amino acids in a polypeptide chain is called the primary structure of that protein. Any change in this primary structure will create a different protein. The secondary structure of protein refers to the shape in which a polypeptide chain can exist. They are found to exist in alpha helix and beta pleated sheet structure. Here, each polypeptide chain twists into a spiral structure called alpha helix. The polypeptide chain forms a right-handed helix. These helices are held together by hydrogen bonds between NH group of each amino acid residue to the CO of an adjacent turn of the helix. In beta pleated structure, all peptide chains are stretched out to nearly maximum. Then they are laid side by side and are held together by intermolecular hydrogen bonds. Here I have compared the primary and secondary structure of proteins. You may visualize tertiary structure of proteins as a further folding of the secondary structure. In fact, it is this structure which gives rise to fibrous and globular shapes of proteins. The secondary and tertiary structures of proteins are stabilized by hydrogen bonds, disulfide linkages, van der Waals and electrostatic forces of attraction. Some proteins are composed of two or more polypeptide chains. These chains are called subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure of protein. Hemoglobin is an example. It consists of 4 protein chains arranged in a specified way. A protein found in a biological system with a unique structure and biological activity is called a native protein. In its native form, when subjected to change in temperature or pH, the hydrogen bonds are disturbed. The globules will unfold and helix get uncoiled and the protein loses its biological activity. This is called denaturation of protein. During denaturation, secondary and tertiary structures of protein are destroyed but primary structure remains intact. The coagulation of egg white on boiling is a common example of denaturation of protein, albumin. With this, we have come to an end of this session. Now, can you tell the reason for curdling of milk? Think about it.
Manorama Horizon. Learning made simple.